My dear students of 11th class, Assalamu alaikum to all of you. And today in your botany, we will look upon the event of plasmolysis. This plasmolysis is in your what we have been studying from the past few lectures, that is the plant-water relations. It is related or other the part of this very chapter, this very topic, that is the plant-water relations, or basically we are studying the transport in the plants. And now this plasmolysis, the behavior of the plant cells or tissues, the behavior of the plant cells or tissues with regard to water movement depends on the surrounding solution, depending upon in which type of the medium the cell is located or what surrounds the cell depends upon that very solution. And the solution can be said to be or said to act as, rather, the solution that surrounds the cell can be said to act as isotonic when the external solution behaves or balances the osmotic pressure of the cytoplasm. It is said to be isotonic. That is, when the external solution balances the osmotic pressure of the cytoplasm. That is, the concentration of the external solution is somewhat similar to that of the internal environment of the cell, and it is said to be isotonic. And it can be either hypotonic if the external solution is more dilute than the cytoplasm. It is said to be hypotonic, and when the external solution is more concentrated, it is hypertonic. So there are three situations or the three types of the media in which the cell could be embedded, or the external environment may be of three types: isotonic, hypotonic, and hypertonic. Isotonic, you must be now knowing when the concentration of the internal and the external environment of the cell is similar. Hypotonic when the external environment has lower concentration, and the hypertonic when the external environment has greater concentration than that of the internal environment of the cell, and the solution is said to be hypertonic there. Now this is to be noted here that cells swell in hypotonic solution due to the absorption of the water. And this is to be noted here that cells swell in hypotonic solution and shrinks in hypertonic solution. Hypertonic solution makes the cells to shrink. This is because of the loss of water. The plasmolysis occurs when water moves out of the cell and the cell membrane of the plant cell shrinks away from its cell wall. When water is lost from the cell and the cell membrane shrinks away from its cell wall, the cell is said to be plasmolysed, and this is called as the plasmolysis. And this occurs when the cell or the tissue is placed in a solution that is hypertonic with more solute to the protoplasm, and water moves out. It is first lost from the cytoplasm and then from the vacuole. The water, when drawn out of the cell through diffusion into the extracellular or outside of the cell fluid, causes the protoplast to shrink away from the walls. The cell is said to be plasmolysed. When the water is lost from the cell, first from the cytoplasm and then from the vacuole, it makes the protoplast to shrink away from the walls, and the cell is said to be plasmolysed. The movement of the water occurs across the membrane, moving from an area of high water potential, that is the cell, to an area of lower water potential, that is outside the cell, in which the cell is in which media the cell is kept or located. The shrunken protoplast is the plasmolysed cell. It is occupied by. When the cell is placed in the isotonic solution, there is no net flow of the water towards the inside or outside. And the external solution balances the osmotic pressure of the cytoplasm. It is said to be isotonic, that means of similar concentration inside and outside the cell, within the cytoplasm and outside the cytoplasm. And when the water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. Such cells are said to be flaccid. The process of plasmolysis is usually reversible. When cells are placed in a hypotonic solution, higher water potential or dilute solution, as compared to the cytoplasm, water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall. This pressure is called as the turgor pressure. And the pressure exerted by the protoplast due to the entry of water against the rigid walls is called the pressure potential. A cell wall is rigid and does not rupture. The turgor pressure is ultimately responsible for enlargement and extension, growth of the cells. And this is the view of the plasmolysed cell, wherein the cell has lost the water. And this is the normal cell, and this is the turgid cell, which occupies more volume than the normal cell because of the absorption of water. Hopefully you must have understood what we mean by the plasmolysis. Thanks. Have a nice day.